Another type of mutation is an inversion. An inversion happens when there is a double splicing of the DNA strand. So it gets spliced here and here. That segment of DNA then kind of floats around, and it can actually turn itself completely around and reinsert itself back into the same spot, only in the reverse order. So here you've got purple on the left and green on the right, and this has just flipped itself over. Now you've got green on the left and purple on the right, and the same sequence, only in reverse order. So what does this do? So basically you've got all of these genes that can be transcribed just fine in either direction. It doesn't make any difference in terms of the protein products, except possibly in these ones in the region of the break. If those were coding regions, those genes might be affected. But inversion basically disrupts this idea of linkage. So again, linkage is the tendency for alleles of different genes to assort together during meiosis, so they're generally connected on the same chromosome. And we saw before that an allele located here is always going to be right next to an allele located here. But then this crossing over broke them up, and so now they're associated with other alleles that were on the other chromosome. But when there are the two homologous chromosomes, and one of them has an inversion and the other one doesn't, they cannot line up in this way during meiosis. The homologous genes are sort of looking for each other to bind together to allow them to line up in this way, and because the genes are now in a different order, they can't bond together and line up during meiosis. So this crossing over is impossible if you've got one chromosome with an inversion and the other one without one. So these inversions tend to cause the genes within the inversion to become locked together, always inherited together with that chromosome, and we call this a supergene. A whole bunch of genes that are inherited together as a unit. And this is fairly common in fruit flies. There's a lot of research showing how locking together beneficial sets of alleles can be a selective advantage in many cases. And so a lot of times these inversions are actually supported and maintained by natural selection. The last two kinds of mutation I want to talk about are chromosome fusion and genome duplication. So both of these two result in a change in the chromosome number. So chromosome fusion is exactly what it sounds like. Two chromosomes get stuck together, become fused together to form one chromosome. And this is extremely rare, but we do have some evidence of it happening in the world, and one of them is from human chromosome 2. So chimps have 24 chromosome pairs. Humans, as you know, have 23. And if you compare the banding patterns of human chromosome 2, they match perfectly with the banding patterns of two chimp chromosomes. So here you have the actual karyotype image, and you can see the banding. And this is diagrammatic form. You can see how these bands on the chromosomes exactly line up between these two separate chimp chromosomes. Not only that, but as you'll recall from Gen Bio and genetics, that there are these structures called telomeres at the ends of chromosomes, and they're associated with particular nucleotide sequences. And those nucleotide sequences that are associated with telomeres are found right in the middle of human chromosome 2, right at the point where these two chromosomes might have joined together to form one chromosome. So, pretty strong evidence that this one chromosome in humans, chromosome 2, is was actually formed by chromosome fusion of these two chromosomes found in chimps and presumably found in the common ancestor of chimps and humans. So because this results in a change in chromosome number, this can allow very, very rapid speciation to occur, because in invertebrates especially, in most animals, you cannot mate with another organism with a different chromosome number. So this is a mechanism for very rapid speciation. Secondly, we have duplications of the whole genome. So this can happen in a couple of different phases of meiosis, either during meiosis 1, if the two homologous pairs don't separate properly, or during meiosis 2, if the two chromatids don't separate properly. You can get a doubling of the number of chromosomes in the gametes, and this can lead to genome duplication. This is actually much, much more common in plants because they're more likely to be able to self-pollinate, and so if they do have a doubling of chromosomes and become tetraploid rather than diploid, they can self-fertilize and so have a mate with the same chromosome number. And this is actually a very well-known mechanism for speciation in plants. It's extremely rare in animals, but there is some evidence that it has, in fact, happened on occasion. And one of those pieces of evidence are these Hox genes. And we mentioned Hox genes when we were talking about molecular homology and evidence that all organisms are joined together in this great tree of life and can be traced back to a common ancestor. Hox genes are a very strong piece of evidence for that because they are found in every single animal species. And they, their sequence, their gene sequences are similar enough that they can be definitively homologized between all animal groups. And so we saw that these Hox genes are present in Drosophila and mouse, and that they can be, that we have homologous sets of Hox genes between those two groups. And we also saw that mice have not one but 4 Hox clusters. So what are these Hox genes? Remember that these are regulatory proteins. They're responsible for turning other genes on and off, determining where in the body they're expressed. They're mainly involved in anterior to posterior body patterning, segment identity, so knowing which segment of the body should produce which structures. And we discovered these Hox genes because of these mutations called homeotic mutations. So homeotic mutations are mutations that involve the wrong body structure appearing in the wrong place. So one of these homeotic mutations is called antennapedia, in which legs grow where the antennae are supposed to be. So basically, wrong information here about what body segment these legs are in. Another example is a homeotic mutation called bithorax. So in normal flies, there's one set of wings and then a very small set of halteres instead of the second set of wings characteristic of insects. And in this bithorax mutation, this first thoracic segment, which normally produces wings, is replicated, so wings are produced instead of halteres in the second thoracic segment. And so, and again, it's an error in location of the correct body segment. So one thing this reminds us of is this fact that every single cell in the body carries all the information that's needed to produce every structure in the whole organism, and what structures develop where simply depends on which genes are turned on and off. And so when these Hox genes are disrupted, we end up with these mutations that are errors of location. So there's very strong evidence that these individual Hox genes in Drosophila are the result of gene duplications due to unequal crossing over. They often have comparable introns They've got their own promoter regions. There's good evidence that that these arose all due to gene duplications. But then the best explanation for this whole scale replication of the entire Hox clusters is a whole genome duplication that happened twice in the gnathostomes. So all gnathostomes have at least 4 Hox clusters. So one whole genome duplication that led to the second set, another whole genome duplication that led to the other 2. So both of these 2 would have been replicated again in the second whole genome replication. And we can actually plot this onto a phylogeny and see exactly where these different things happened. So outgroup, we can consider this Drosophila has 1 Hox cluster, so we can be sure that that is the plesiomorphic condition, the ancestral condition. Cephalochordates, which are close relatives of vertebrates, but they're not vertebrates, also have a single Hox cluster. Lampreys, which we saw are vertebrates but not gnathostomes. By the time lampreys branched off, there had been 1 whole genome duplication, so they have 2 Hox clusters, and somewhere on the line leading to gnathostomes or the jawed vertebrates, there was another duplication event that led to the 4 Hox clusters. And so all tetrapods have 4 Hox clusters. Interestingly, actinopterygians, or the ray-finned fish, have a highly variable number of Hox clusters, going anywhere up to 8, which we see in pufferfish. And so probably what happened is that there was an additional whole genome duplication in the line leading to actinopterygians with subsequent deletions of Hox clusters in different groups within those fish. So our discussion of all of these different types of mutation lends itself well to sort of a nice categorization of the different kinds of mutations. So we saw that point mutations and insertions and deletions lead to new alleles, so different forms of the same gene. Gene duplication and genome duplication both lead to an absolute increase in the number of genes. So gene duplication is just going to increase the number of genes by 1. Whole genome duplication is going to completely double the amount of genetic information there. And this is extremely important to evolution, as we will see and discuss in the next class. We saw that chromosome fusion and genome duplication can result in very rapid speciation because it's essentially an instant change in the number of chromosomes. And then we saw that these inversions increase linkage and create supergenes, even though most likely they don't result in any kind of change in phenotype because the same genes are still present.